Hey guys, what's good? So, what are biomolecules? Biomolecules are organic molecules that a living organism produces. They are essential for the survival of cellular organisms and perform a vast array of functions. There are four main categories: carbohydrates, lipids, proteins, and nucleic acids. You might have heard of them in conversations about diet, health, or in the form of food, because that's where their presence is most easily observed. Recognizing these molecules is key to understanding biochemistry. So, to start off, something you need to know is that most biomolecules have some sort of variant of a monomer, a dimer, and a polymer. So, what's a monomer? Say we're building a Lego minifigure. Each individual brick that makes up the minifigure would be known as a monomer. A monomer is a building block that makes up upon a more complex structure. Numerically, it's like the unit 1. So when two monomers bind, they form a dimer. A dimer can numerically be thought of as the number 2, and they highlight an important concept to understand about biomolecules: bonding. The bonding process is unique to each biomolecule. Polymers are formed from many monomers binding together. They are the large biological structures that monomers form and are able to serve many functions. An example of a polymer is DNA. So starting with carbs, they are the most abundant biomolecule on Earth. There are high concentrations of them in potatoes, pasta, bread, and honey. Carbs are the immediate source of energy for cells, tissues, and organs due to their high energy concentration. The body breaks carbs into glucose used instantly for bodily functions or transformed into glycogen, which can then be stored in the liver and muscle cells for later use. Carbohydrates have many monomers. These monomers are called monosaccharides, or simple sugars, and come in many forms. Some examples are glucose, fructose, galactose, ribose, and deoxyribose. Carbohydrates dimers, or disaccharides, include sucrose, lactose, and maltose. Wait, but how are disaccharides formed? Disaccharides are formed in a process called dehydration synthesis, which links two monosaccharides together. The structural composition of monosaccharides have an OH at the bonding site. An oxygen and hydrogen paired together is called a hydroxyl group. In dehydration synthesis, a hydroxyl group and a hydrogen atom are removed to create a glycosidic bond between the monosaccharides, making them disaccharides. Remember, the hydroxyl group is an oxygen and a hydrogen. So when a hydroxyl group and a hydrogen atom are removed, we see that combined, an H2O molecule was removed, also known as water. This explains the name dehydration synthesis. D, which refers to away or off, hydration, which refers to water, and synthesis, which means the combination of elements to create a connected whole. With dehydration synthesis, even the name can tell you what is going on in the process. Carbs polymers are called polysaccharides, which are hundreds to thousands of monosaccharides bonded together in dehydration reactions. These large strands of monosaccharides form substantial structures. Let's focus on 3. Starch is a polysaccharide made from links of glucose that plants use to store energy. Glycogen is the polysaccharide made of links of glucose used by animals that's stored in the liver and muscle cells for later use. And cellulose is the most abundant organic compound on Earth and is made of alternating glucose links. Animals do not possess the enzymes to hydrolyze cellulose or process it, and humans can't digest it, but they do help our digestive system in the form of fiber. Cellulose is so durable because it's reinforced by hydrogen bonding on the alternating glucose links. Moving on from carbs, I'm now going to talk about lipids. Lipids are a diverse group of molecules that all share one common trait: they don't mix well with water. Hence, they are called hydrophobic. There are 3 categories that we're going to focus on: fats, phospholipids, and steroids. Fats, also called triglycerides, are made of 2 components: a glycerol molecule and a fatty acid tail. Glycerol and fatty acids bond through dehydration synthesis and create an ester bond. The fatty acid is composed of a carbon chain that is by default connected through single bonds. However, when a carbon chain contains one or more double bonds, it's called an unsaturated fatty acid, while one with no double bonds is called a saturated fatty acid. This is because when a double bond occurs, the carbon chain will lose a hydrogen. Therefore, it's not saturated with the maximum number of hydrogens that it could possibly have. If a carbon chain has one double bond, it's called monounsaturated, and if it has multiple double bonds, it's called polyunsaturated. If at room temperature and in the form of liquid, these are called oils, and oils are known for being nonpolar or hydrophobic. Water, however, is extremely polar, creating unique behaviors and interactions. Polar substances can dissolve other polar compounds or ionic substances, like salt, but they can't dissolve nonpolar substances. Oils and all lipids cannot mix well with water because they have molecules on them that characterize them as nonpolar. While the term hydrophobic may sound like a negative one, it's actually quite helpful. For example, our next lipid utilizes this to create cell membranes. Phospholipids are like triglycerides, except they only have two fatty acid tails, which can be saturated, unsaturated, or both. Their name derives from the fact that they have a phosphate group attached to the glycerol molecule, phospholipid. The phospholipids make up the cell membrane in a structure called a lipid bilayer, with the head of the phospholipid facing outwards to the cell and the tails facing inwards. This is because the fatty acid tails of the phospholipids are hydrophobic and want to stay away from the water, while the heads are hydrophilic, wanting to stay near the water and forming hydrogen bonds. It's called a bilayer as there is another layer of phospholipids in the opposite orientation to the exterior layer. The structural integrity of a cell membrane is called membrane fluidity, and it's influenced by the surrounding temperature. When it's too hot, the membrane becomes fluid, while when it's too cold, the membrane becomes rigid. This is harmful to the cells and prevents the exchange of nutrients across the cell membrane. To counter this, biology evolved to integrate cholesterol in the cell membrane to prevent both extremes. Cholesterol makes animal cell membranes more fluid during the cold temperature and more stable at hot temperatures. So basically, cholesterol upholds the integrity of the cell membrane structure, and cholesterol is a steroid. Another biomolecule that has a wide range of functions are proteins. Before we get into them, know that function is dependent on structure. This idea is present throughout all of biochemistry, but it's especially prominent in proteins, so make sure you know that. A protein is a polymer that serves as the language of life. Proteins can transport molecules, act as structures or storage, or as enzymes that expedite biological reactions. Proteins do almost everything. The monomer of proteins is called an amino acid. An amino acid is composed of an amino group and a carboxyl group alongside an R group. The R group is a substitute for whatever will actually be in its place, and it can be either hydrophilic or hydrophobic, which will influence the protein's interactions with other proteins. Hydrophilic R groups can be basic, acidic, uncharged, or polar, and hydrophobic R groups will be nonpolar. In water, amino acids can become ionized. What this means is that the carboxyl group will lose a hydrogen atom, and the amino group will gain a hydrogen atom. The dimer of a protein is called a dipeptide and is formed from a dehydration reaction between two amino acids. The reaction occurs between the carboxyl and amino group and forms a peptide bond. Proteins in and of itself are a polymer, but a more accurate term would be a polypeptide. A polypeptide structure can be tracked in 4 degrees of levels. First, there's a primary structure, which is a polypeptide chain formed by peptide bonds. You'd find a primary structure like this coming from a working ribosome in the cell. The secondary structure is characterized by the polypeptide links taking a structural form. A secondary structure can either be called a alpha helix or a beta pleated sheet. These structures are formed from interactions of hydrogen bonding. The tertiary structure is achieved when ionic bonds, disulfide bridges, and or hydrophobic interactions stabilize the structure. Those bonds are dependent on the R group's properties. Now, depending on its purpose, a tertiary level of structure can be fully functional. Now, the R groups, or side chains, are characterized by their hydrophobic or hydrophilic nature, right? But when placed in an aqueous environment, unique interactions occur. The hydrophobic side chains fold inwards, while the hydrophilic side chains fold outwards and create hydrogen bonds with the water. An example of this is the phospholipid bilayer of a cell membrane. The final level of a protein shape is the quaternary structure. Here, multiple polypeptide chains act as subunits and bond together to create a fully functional protein. Hydrophobic and hydrophilic reactions also influence how the different polypeptides form. An example of a quaternary structure would be hemoglobin, the protein that carries our oxygen in the bloodstream, which is composed of 4 tertiary structures combined. Denaturation. Proteins can lose their quaternary, tertiary, or secondary levels of structure by chemical means like heat or radiation. While they maintain their peptide bonds, they lose their structure, inadvertently losing their function as well. Remember, structure is function. An increase in heat or chemical radiation would increase the molecular movement between the side chains, disrupting the bonds that gave it its original shape. Denaturation is often permanent and not reversible. An example of it would be fried eggs, as the egg whites were once clear prior to being heated up in a process of denaturation and are now, egg white. The final biomolecule that you need to know are nucleic acids. You may recognize them as DNA or RNA, which are the two forms of polymers that nucleic acids can take form as. Nucleic acids are located in the nucleus and are responsible for genetic inheritance. They are involved in storage, transfer, and the expression of hereditary information. RNA's role is to assemble proteins, as we just discussed, according to the DNA's instructions. DNA carries instruction for its replication and has the code to make proteins. DNA resides within the chromosomes. The monomers of nucleic acids are called nucleotides and are made of three components: a phosphate group, a sugar, and a nitrogenous base. Together, the phosphate group and a sugar create a backbone called the sugar-phosphate backbone that is connected via dehydration reactions between the sugar and the phosphate group. The nitrogenous bases are similar to the R group discussed for proteins, as the term is a placeholder for the specific base. The base can be adenine, guanine, cytosine, thymine, or uracil in the case of RNA. RNA versus DNA. While similar, both RNA and DNA serve different purposes and are composed differently. Both have a phosphate group, but they have different types of sugars. DNA has deoxyribose, while RNA has ribose. While DNA can have adenine, guanine, cytosine, or thymine as potential bases, RNA can have adenine, guanine, cytosine, or uracil as potential bases, replacing the thymine. In terms of structure, DNA is a double helix while RNA is a single nucleotide strand. This creates specific interactions between the nitrogenous bases and DNA, as its structural shape is a double helix. Adenine will always pair with thymine, and cytosine will always pair with guanine. The strands are complementary, making them predictable each other. The nitrogenous bases complementarily bond via hydrogen bonds in DNA. Another thing to note is that DNA can lose its double helix due to denaturation, similar to proteins. That's a brief overview of the four central biomolecules. I hope you learned something, and see you later. Subscribe.